Neurofibromatosis. The cell signaling process helps ensure that crucial activities occur in the right cells at the right time and in proper coordination with the activities of other cells of the organism. Reception is the target cell's detection of a signaling molecule coming from the outside of the cell. A chemical signal is detected when the signaling molecule binds to a receptor protein located at the cell surface, or, in some cases, inside the cell. Transduction is a step or series of steps that converts the signal to a form that could bring about a specific cellular response. Transduction usually requires a sequence of changes in a series of different molecules, a signal transduction pathway. In the third stage of cell signaling, the transduced signal finally triggers a specific cellular response. Regularly in reception, when the appropriate signaling molecule binds to the extracellular side of the receptor is activated and changes shape. Its cytoplasmic side then binds and activates a G protein. The activated G protein carries a GTP molecule. The activated G protein leaves the receptor, diffuses along the membrane, and then binds to an enzyme, altering the enzyme's shape and activity. Once activated, the enzyme can trigger the next step, leading to a cellular response. Binding of signaling molecules is reversible. The activating change in the GPCR, as well as the changes in the G protein and enzyme, are only temporary. These molecules soon become available for use. Neurofibromatosis is a genetic disease characterized by light brown spots on the skin and neurofibromas, small benign growths under the skin, among other signs. Approximately 1 in 3,000 people worldwide are affected. As it is a genetic disorder, only treatment is available at the moment, not a full cure. It is a dominant autosomal disease and is passed down from parent to child most of the time. Sometimes, however, the condition appears to occur spontaneously in the initial sperm and/or egg cells. The disease ranges in severity, largely based on type and severity of the genetic mutation. Neurofibromatosis is caused by a mutation in the genetic sequence. Specifically, there is a mutation in the gene 17q11.2 on chromosome 17, or the NF1 gene. The gene codes for the creation of the neurofibromin 1 protein. This protein is directly related to the RAS cell signaling pathway, which serves to regulate cell population growth, among other things. The NF1 protein is a negative regulator for this pathway, i.e., when it is activated, the pathway is signaled to slow cell proliferation. Mutation of the gene means that NF1 proteins created are misshapen and fail to receive proper signals. The response to the signal is transcription, the synthesis of mRNA, which will be translated in the cytoplasm into a specific protein. We have seen that, in a healthy human being, neurofibromin 1 receives a signal to slow cell population growth from a G protein-coupled receptor and causes a negative response. In someone with neurofibromatosis 1, the misshapen NF1 protein fails to communicate properly with the GPCR in the RAS signaling pathway. Therefore, cell proliferation continues perpetually, resulting in overgrowth. Flat, light brown spots on the skin, having more than 6, occur prior to birth or in the early years of life. Freckling in the armpits and/or groin usually appears by age 3. Soft bumps on or under the skin, the beginning of tumors. Tumor on the optic nerve usually appear by age 3. Learning disabilities, impaired thinking skills. ADHD is also common. Short stature, below the average height.